Thank you for Professor Yang's introduction. And I, I'm I'm Chao Chen from China Medical University. And first, I want to thank the committee to invite me to present my research work here. Today, my topic is mass spectrometry-based screen of kinase activity and active components in Chinese medicine. Because I'm in charge of proteomics collect in CMU and CMUH, so we can offer some service and some cooperation activities, and which includes the identification and the quantitation of proteins and the small molecules within the metabolites. And my research topic also interested in the discovery of protein and metabolite biomarkers by proteomics and metabolomics approaches. And we also try to build the platform. This platform is talk about the large-scale analysis of protein kinase activity. And recently, we also built a new research area is to screen active components from Chinese medicine. So today, my topics will include these two areas. So first, we want to use the large-scale kinase activity platform to discover new mechanisms of Taxol in treating ovarian cancer. Taxol is very famous anti-cancer drug. So we take this as an example to test our platform to see if we can discover new mechanisms. As many of you already know that the protein phosphorylation is very important in signal transduction, cell metabolism, and apoptosis. So actually, protein kinase are very important target for drug discoveries. And however, in the traditional methods to detect the protein kinase is by the radiometric-based assay. However, this assay is restricted by its use of radioisotope materials. It have safety concern issues. And other approaches such as antibody-based or peptide array, they are rely on the specificity of antibodies. So sometimes they will have some problems about the low specificity and the tedious procedure problem. So we are thinking about if we can use the MS to identify the phosphorylation proteins. And so some of you may heard about the phosphoproteomics, right? And phosphoproteomics is very arise about 10 years ago, so and have become a very huge research area in mass spectrometry. However, the content of phosphopeptides relative role in mixture of peptides, and also phosphopeptides are usually unsuppressed by the presence of non-phosphorylated peptides. So before we want to detect the phosphoproteins, we have to use some materials such as TiO2 to enrich phosphopeptides, or you have to use a multi-dimensional method to fractionation complex peptides to increase the detection sensitivity, or you can purchase more advanced LC system. It's very, very, very expensive. So not everyone can afford the very expensive instrument. So we are thinking another approach is to use the chemically synthesized peptide substrate. Actually, this principle is just like you use the peptide array, but because peptide array, the detection of peptide arrays rely on the antibody, right? But we transfer this concept to use the MS to detect the peptide array. So compared to the intact protein substrate, peptide substrate are relatively expensive to synthesize and easy to store, handle, and catalyze. And also we can use the monitor, another kind of MS. We can use to do the high throughput screen of the phosphopeptides. So we synthesize the 38 peptide substrate, and this 38 peptide substrate can also represent major kinase activities here. This is our workflow to discover new mechanism of Taxol in treating of ovarian cancer. And this workflow actually is a little complicated. But first, we can prepare the cell lysis, and we just add the peptide substrate into the cell lysis. So the peptide substrate will be phosphorylated protein to its kinase activity. Then we purify the peptide substrate by the TiO2 and the solid phase extraction device. Then we use the narrow LC MSMS to detect the phosphorylated peptides, and use the bioinformatics software to identify them and quantify them. Then in the final step, we can use the Western blot to verify the result. So which means that we can further confirm the specificity of this peptide substrate to the kinase. Okay, because we also use the Western to verify the results. And so we can also use the bioinformatics software to highlight the major mechanism in the kinase activities. And also we can find a new kinase they are not reported before in the treatment of ovarian cancer cell line by Taxol. So we can, shown here, we find that the AKT, SGK3, IKK beta, and PK, they are new kinase in the treatment. So we can use the kinase inhibitor to enhance the Taxol treatment for the ovarian cancer cell line. So and the second topic is the we can also to use the MS system to screen the active compounds from transmedicine. So we take the SARS-CoV-2 virus as an example. In the infection of SARS-CoV-2 to the host cells, they are rely on the SE2 protein in the host cell and the RBD protein of the SARS-CoV-2. So once they are binding, this virus can get into the host cell and make the infection. So we are thinking about how to design this experiment to screen the active components. So we use them. What we use is a magnetic beads and to bind with the SE2 protein, and we can use the reaction is the RBD, and this RBD can interact with the SE2, right? So if in control group, if there are no compound to inhibit their binding, actually when we elute these two proteins out and to do the choosing digestion and followed by the multi-top analysis, we can find the peptide patterns such as like this one. But if there is a compound that can inhibit the bindings between these two proteins, then we can observe this pattern. So we can quickly screen out what kind of compound can inhibit their binding force. Then we can use the LC system to identify the compounds in the transmitters. We have screened 100 traditional transmitters, and then finally there are 5 transmitters, and they have an inhibitor efficiency for their bindings. And we also further identify their compounds inside And we also purchase the standard and the test again to further confirm if this compound can actually inhibit their bindings. So there are 3 compounds we find. And we also use a molecular docking to check their energy, okay? And finally, our ongoing future project is to still continue discover new mechanisms for disease and drug treatment, and still rapid screen of bacteria and its antibiotic resistance. And what we want to do is to build up a TCM-based compound library. It's a very huge work, and we are continuing right now, and to still discovering active components to treat disease. Okay, and thanks for everyone. Thank you.